afternoon, everyone. Thank you for joining us for this webinar today. I'm Cassie, Marketing Supervisor at GenScript, and I'll be the moderator for the webinar today. So a little background about GenScript for those who are new to us. GenScript is the leading life science research and application service and products provider in various fields, from basic life science research to translational biomedical development, industrial synthetic products, and cell therapeutic solutions. The topic for today's webinar is cPass SARS-CoV-2 Neutralizing Antibody test for post-vaccination monitoring, and we have our field application scientist, Dr. Chengxun, to present for today. Dr. Su received a PhD training in biomedical engineering interdisciplinary graduate school at Nanyang Technological University, Singapore. During her PhD, she focused on developing bioengineered platforms for disease modeling and drug testing applications, where she applied cutting-edge technologies in cell biology, microfabrication, microfluidics, and molecular biology to develop novel tools to support fundamental and translational research. Using these cutting-edge technologies, Dr. Su spearheaded several collaborations with the clinicians to investigate immune dysfunction and inflammatory profile in patients who recovered from COVID-19, paving the way for potential intervention strategies. This webinar will take around 45 minutes and we'll have about 15 minutes for Q&A session. You can type in your questions in the chat box and we'll answer them after the presentation. If we didn't get to answer your questions during the Q&A session, we'll email the answers back to you after this webinar. So without further ado, we will let Dr. Su to start her presentation now. Dr. Su, over to you now.
Thank you, Cassie. So thank you very much for joining today's webinar. Today, I will be presenting on the cPass SARS-CoV-2 Neutralizing Antibody test for post-vaccination monitoring. So the agenda for today's presentation. So first, I will introduce protective immunity and neutralizing antibodies. Next, I will introduce the surrogate virus neutralization test, which is the technology behind cPass. I will cover topics including neutralizing antibody as the correlate of protection and detection of neutralizing antibody against variant concerns using the SVNT, as well as how it can be applied to develop the next generation of vaccine. Lastly, I will also discuss the dynamics of the neutralizing antibody response, including the durability of neutralizing antibody, as well as the possibility of adopting a neutralizing antibody-guided booster dose regimen. First, I would like to introduce the mechanism of infection for the SARS-CoV-2. When the virus enters the human body and comes into contact with the cells, the spike protein on the surface of the virus binds to the ACE2 receptor on the cell, which then triggers the internalization of the virus into the host cell. Following this, the virus will replicate and cause illness. When we take a closer look at the protein-protein interaction, it was found that the receptor binding domain on the spike protein, abbreviated as S1RBD, binds specifically to the ACE2 receptor. So when we are vaccinated or infected, our body develops adaptive immunity that will protect us from future infection or severe diseases. Adaptive immunity consists of humoral immunity and cell-mediated immunity. Elimination of infected cells is mediated by T cells, which are activated after the virus enters the host cell, and it can kill the infected cell and stop the viral replication. Hence, it is very important in protection against severe disease. On the other hand, the prevention of virus-host cell interaction is mediated by antibodies. Based on the function of antibodies, we can classify them into binding antibody, which binds to virus but may not block the virus-host cell interaction, and also neutralizing antibody that is shown in this image here, that binds to the RBD region on the spike protein and block the interaction with ACE2 receptor, thus inhibit the cellular entry of the virus. Because neutralizing antibody acts before the infection takes place, it plays a very important role in preventing transmission and spreading of the virus. I would also like to share from the recent update on adaptive immunity and SARS-CoV-2 from CDC. As shown here, the immunity is a gradient, and T cells are main contributors to prevent hospitalization and severe diseases, and antibodies, including antibody location, functionality, isotype, and antibody levels, are more important in preventing mild disease, and also it can confer sterilizing immunity. As shown in their concluding slide, neutralizing antibodies likely confer a majority, but not all, immunity. In recognizing neutralizing antibody is the fraction of antibody that confers functional immunity, FDA has issued a series of guidelines regarding use of neutralizing antibody to fight against this pandemic. Due to its functionality to block virus entry into host cell, neutralizing antibody has been authorized as COVID therapeutics. It can be either obtained from convalescent plasma, or it can be obtained by recombinant expression. Importantly, the plasma obtained from recovered COVID patients must have high neutralization antibody titer to be qualified as convalescent plasma. Apart from use as a therapeutics, assessment of neutralizing antibody titer is also required to determine the vaccine effectiveness. As shown here, according to the FDA's guideline on development and licensure of vaccines to prevent COVID-19, the functional activity of immune response, which is mediated by the neutralizing antibody, need to be evaluated in vitro. So in the most recent version of the Moderna and Pfizer vaccine fact sheet, the effectiveness of the booster dose was based on assessment of the neutralizing antibody titer. And for the Pfizer vaccine, the immunogenicity of the primary series in adolescents between 12 to 15 years old was also based on assessment of neutralizing antibody. So how is neutralizing antibody titer determined? Conventionally, the virus are mixed with patient serum, which may or may not contain neutralizing antibody. Following that, the mixture was incubated with live cells. As shown here, for samples does not contain any neutralizing antibody, the virus will infect the cells. While for sample that contains neutralizing antibody, the antibody will block the virus entry into the cell, thus reducing the number of infected cells. So by quantifying the amount of infected cells or uninfected cells, the neutralizing antibody titer can be determined. The advantage of this assay is that it is gold standard in specificity in detecting the functional neutralizing antibody. However, due to the highly contagious virus used, it requires strict safety conditions, biosafety safety level 3. As shown in this image here, the operator will need to have a PPE including full body jumpsuit and also respirators. The assay is also tedious and slow, can take up to 3 to 5 days to obtain the result. And due to the complexity of the assay, it requires highly skilled operators. The assay is also very expensive and not scalable, thus not suitable for high throughput applications. And because of the protocol and material differences used between different labs, the results are often not comparable, obtained from different labs. So to overcome these technical challenges, a surrogate virus neutralization test, abbreviated as the SVNT, was invented, which allows for highly specific detection of neutralizing antibody in a much more user-friendly manner. As I mentioned earlier, the neutralizing antibody binds to the RBD region on spike protein to block its interaction with the ACE2 receptor. So based on this mechanism, the surrogate SVNT use purified RBD from the S protein and the host cell receptor ACE2 to mimic the virus host cell interaction in an ELISA plate well. As shown in this schematic here, the RBD used in SVNT is conjugated with HRP, And once the RBD binds to the ACE2 receptor coated on the plate, it will show a colored signal. And when there's neutralizing antibody present in the sample, it will form a complex with the RBD and subsequently washed away in the washing steps, thus showing a much reduced colored signal. This assay is much more user-friendly and can be used in a biosafety level 2, and the assay can be completed in 1 to 2 hours, and is also automatable and more suitable for high-throughput applications and mass testing. And because of the standard protocol we use, the between-lab inconsistency is minimized. The assay is also highly specific in detection of all functional neutralizing antibodies that can block the RBD ACE2 binding. And the detection is not limited to certain isotypes such as IgG or IgM. And the RBD in the SVNT is in the solution instead of coating on the plate. So this allows the RBD to be in a more native conformation that more closely resemble the in vivo condition. So how comparable is the results obtained from SVNT versus the gold standard left virus neutralization test? As shown here, the results obtained from the SVNT and the PRNT, which is the plate reduction neutralizing test, using the live virus assay. So we can see that the results are highly correlated with the R-squared value equals to 0.945, and this high correlation is considered very high for any biological system. On the other hand, for other commercial anti-SARS-CoV-2 serology assays that is designed to detect binding antibody, their correlation with the gold standard live virus test is very poor, with R-squared value less than 0.5. Additionally, we have also validated the assay using a total of 140 serum samples collected from SARS-CoV-2 RT-PCR positive and negative individuals. As shown here, the clinical agreement using the PRNT50 as the comparator shows that we have a very good sensitivity and specificity with 100% for both the positive agreement and negative agreement. For the blood collection to run the test, because we only require very little amount of the sample, we can either use finger prick or venipuncture to collect the blood sample. Here we also show 100% agreement for the two methods. So due to its high specificity and sensitivity in detection functional neutralizing antibodies, the cPass SVNT test was authorized as the first test that detects neutralizing antibody against SARS-CoV-2 by the FDA EUA. And subsequently, we have registered the cPass for in vitro diagnostic use in more than 10 countries, including the USA, Europe, Singapore, Brazil, United Arab, and Argentina, etc. So apart from being used as a research tool, the cPass can also be used as a in vitro diagnostic tool for people to walk into the clinic to order the test. Next, I'm going to introduce how to interpret the cPass reading. So the level of the neutralizing antibody is calculated from the OD value, and the results can be expressed as the percentage inhibition as shown in the formula here. The higher the percentage inhibition, the higher level of neutralizing antibody. So the cutoff value for differentiating positive and negative sample is 30% based on validation with our panel of confirmed vaccinated patient plasma and no vaccinated control plasma, as well as correlation with live virus neutralizing test. Alternatively, the user can also choose to use the neutralizing antibody standard to obtain results on the neutralizing antibody concentration. As shown here, you can obtain the standard curve using the calibrator and interpolate the concentration for your sample based on their OD value. What we have as an additional tool is the International Standard of Neutralizing Antibody from WHO that allows for calibration and harmonization of the serological assays detecting neutralizing antibodies. As you can see, for previous cases, the hepatitis B, 10 IU per liter is considered a protective level of antibody. You know, for COVID-19, the WHO has established the international standard for neutralizing antibody, and of particular note, the IU per ml is reserved for neutralizing antibody assay, and because cPass is one of the assay that detects functional neutralizing antibody, we will be able to report the results in IU per ml. And for binding antibody assays, they can report their results as binding antibody units, BAB per ml, BAU per ml. So many of you are interested to know whether we can directly convert the SVNT percentage inhibition to IU per ml. To investigate this, a collaborative study was conducted across 3 regions, including China, Monaco, and Singapore, to study the correlation between the percentage inhibition and the international standard. As shown here, using the international standard as the calibrator, we observed a very consistent trend of the conversion between percentage inhibition and the IU per ml across different regions. So this is the curve after averaging the results, and we see a very high R-squared value of 0.977. So currently this work has been accepted in principle, and we hope to see it published soon so that the cPass users can directly convert the percentage inhibition into IU per ml. In addition to FDA's guideline on assessment of neutralizing antibody to determine the effectiveness of the vaccine, several thought leaders in the field have also recognized neutralizing antibody as a good predictor of protective immunity. Here, I would like to share the quote from Johns Hopkins University stating that ultimately, neutralization assays are the gold standard for determining if a patient has effective antibodies and protective immunity against SARS-CoV-2. And Dr. Anthony Fauci, who has always been advocating that neutralizing antibodies are the gold standard for protective immunity. Additionally, I would also like to share some of the key takeaways from the recent WHO R&D blueprint consultation, where experts meet to discuss the emerging evidence towards the establishment of correlates of protection. As stated by experts in the field, there is growing evidence showing that neutralizing antibody responses could be useful in predicting vaccine-induced prediction. And also, as stated by another expert, the simplest option for any vaccine development is high-level, long-lasting neutralizing antibodies. Next, I would like to share some of the key scientific evidence that shows that neutralizing antibody is a good correlate of protection. In this recent publication in Nature Medicine, the relationship between in vitro neutralization levels and the observed protection from infection was analyzed using data from 7 current vaccines and also convalescent cohorts. The vaccine study include the mRNA vaccines, the adenovirus vector vaccines, and also the inactivated virus vaccines. As shown from their result, the neutralization level and the protective efficacy is highly correlated to each other. Another study, which is the Moderna phase 3 efficacy trial, also reported similar findings. As shown in this graph here, the higher the neutralizing antibody, the higher the vaccine efficacy observed. And some key numbers to take note is that the vaccine efficacy is estimated to be 45 to 60% for vaccine recipients without any detectable binding or neutralizing antibodies. And the vaccine efficacy increased to more than 98% for recipients with the highest neutralization titers. I would also like to share a study that described an outbreak of SARS-CoV-2 on the facial vessel associated with a very high attack rate. As shown in this study, the presence of neutralizing antibodies from prior infection was significantly associated with protection against reinfection. As shown in this table here, for all the 6 crew members, so all of them have tested positive using the Abbott binding antibody assay. However, only those who are tested positive for the cPass assay are protected from infection. Another real-life case study in Israel further confirmed the correlation of neutralizing antibody level with occurrence of breakthrough infection among the vaccinated. So these are the neutralizing antibody titers determined for the breakthrough infection group and also the case control group. And we can see that the case control group have a much higher neutralizing antibody titer. So in this study, the vaccine used is the Pfizer mRNA vaccine, and the overall efficacy against for prevention of infection and mild disease dropped from 94.3% to 64% over 1 month. And this drastic drop of vaccine efficacy is mainly due to the waning immunity and as well as the emergence of variant of concerns. As we know, current vaccines are based on the wild-type strain. So for variants that have amino acid change near the spike receptor binding domain, it could result in reduction in neutralizing titers. So according to the summary from CDC, there's a neutralization loss that range from 0 for alpha to about 7-fold for the beta strain. And the delta strain is approximately 1.5 to 2-fold reduction in neutralization activity. And this study that's recently published in Nature investigated the neutralization of the wild-type and variant virus by the Pfizer-induced immunity. So as shown in the results here, the neutralization level against all the variants have dropped, and we observe a most significant drop for the Kappa strain. So this means that although there's a drop in the neutralization activity, there's still a protection from the vaccination, although at a lower level. So I believe many of you are wondering whether our SVNT can be used to detect neutralizing antibody against variant. And yes, we can do so by replacing the wild-type RBD to variant RBD. In this graph, as indicated here, that by simply replacing the blue region from the wild type to the variant RBD, we can detect neutralizing antibody against variant. So this is the list of all the variants available for purchase. So using our SVNT with the variant RBD, a study from Singapore shows that for patients who was previously infected with the wild type strain, their neutralization against the wild type is the highest and drops slightly for the alpha strain, and the reduction in activity against the beta is more significant. For patients who are previously infected with the alpha variant, it was observed that their neutralization activity against alpha and wild type is still quite high, and there's a loss of neutralization against the beta strain, and vice versa for patients who are infected with the beta strain. So another study from USA also reported similar findings. As shown here, for patients who have been infected with the wild type, we observe that their neutralization activity against the wild type is the highest across all the 3 different level of neutralizing antibody. So we also observe that the protection against the Brazil is the lowest for all the 3 groups. You know, when Delta variant becoming the prevalent variant of concerns for the last few months in Singapore, we also performed an internal study to investigate the difference between neutralization against the wild type and the Delta variant. So cPass stands for the COVID pass, and dPass stands for the Delta version of the COVID pass. So as shown here, there's still a very high correlation between the cPass results and dPass results with the R-squared value equals to 0.8. And well, generally, we observed a reduction of the dPass results that range from 5% to about 20%. So this is the averaged value. So in average, we observe about a 15% drop for the dPass reading. Another study from USA also used the SVNT to evaluate the neutralization activity against variant of concerns among the vaccinated. As shown here, for people who are vaccinated with Pfizer or Moderna, their neutralization against the beta and alpha dropped a little bit compared to the wild type. However, for people who are vaccinated with the Johnson & Johnson vaccine, their neutralization level against the beta and alpha dropped significantly to almost negative. A study from Singapore investigated for people who have taken the Pfizer vaccine. The study shows that their inhibition level against the wild type and the alpha still remains high, but there's a significant drop when it comes to the beta variant. So the next question you may have is that whether binding antibody assay can differentiate differentiated between the variant of concerns. As shown in this comparative study, the SVNT showed lower level of inhibitory effect against the alpha and beta variants, while the binding antibody assay does not show any difference when tested with the variant RBDs. This means that a very high binding antibody level can actually give a false signal that suggests high protection, while in reality, the functional immunity against the variant may have dropped significantly. So this is a good mechanistic explanation on why binding antibody assay may not show the differentiation, while neutralizing antibody assay is able to. So as shown here, we have generated 4 antibodies that have strong binding activity to the receptor binding domain. And out of these 4, only 2 showed neutralization activity. And these 2 are not necessarily the strongest binders. So this can also explain that sometimes for people who have taken both neutralization test and also the binding antibody type test, there are cases where their binding antibody level is high, but their neutralizing antibody level is low. Next, I would like to discuss how SVNT can be used as a tool for vaccine development. So the first generation of vaccine is based on the SARS-CoV-2 wild type, and it offers the highest protection against the wild type virus. And it is of urgent need to develop the second generation of vaccine that can offer very high protection against all the existing SARS-CoV-2 variants. And it is also important to look at the development of the third generation vaccine that can not only protect us against existing variants and also other SARS-related coronaviruses, which may likely cause the next pandemic. In a recent study by the cPass inventor, they investigated the vaccine efficacy not only against the SARS-CoV-2 clade and also the SARS-CoV-1 clade. So for the SARS-CoV-2 clade, they look at the efficacy against the wild type and also the alpha, delta, beta variant, and also 3 pre-emergent subunit viruses, which are now in the pangolin and bat, and may one day transmit into human. And they also look at 3 SARS-CoV-2 related viruses, and also include 2 of them is also pre-emergent, which are now still in the animal. So this study was performed using the SVNT technology because for the pre-emergent subeco viruses, the live virus are not available. That means that it is impossible to conduct such study using the live virus assay. So in this scenario, the SVNT can be a very powerful tool to allow the detection of neutralizing antibody against these pre-emergent subeco viruses simply by using the receptor binding domain for these viruses. Their results shows that For healthy people who received the Pfizer vaccine, their protection against the SARS-CoV-2 correlate it remains high, with the slight drop in the beta variant, and also the protection against one of the pre-emergent virus is slightly lower. And their protection against the SARS-CoV-1 virus is at a very low level. And for patients who have been infected with the COVID and then received vaccine, their protection against all the SARS-CoV-2 clade are very high. However, their protection against SARS-CoV-1 is slightly lower, although it's at a higher level as compared to people who have not been infected before. This is a very interesting result that for patients who have been infected with the SARS-CoV-1, which is responsible for the SARS outbreak in 2003, when they received the Pfizer vaccine, they are not only protected from the SARS-CoV-2 clade and also the SARS-CoV-1 clade. So this means that there's high level and broad spectrum of neutralizing antibody for SARS survivor who are vaccinated with the COVID-19 vaccine. So this actually offers a very promising insight in developing the third generation of vaccine. So for SARS survivor who are vaccinated with the SARS-CoV-2 vaccine, they have very high neutralization level against both clades. So how about we reverse the order? You know, for the people who have received the SARS-CoV-1 vaccine, maybe we can base on the SARS-CoV-1 sequence and to develop the next vaccine. So from this study, it is shown that the SVNT can be a very powerful tool for vaccine development, especially for viruses who are pre-emergent, where the live virus is not available to conduct experiment. As many of you are wondering, how long can neutralizing antibody last in our body? And does everyone react the same after infection or vaccination? So this is a very important evidence obtained from a cohort of recovered patients. So as shown in the graph here, the neutralizing antibody response can be classified into these 5 distinct groups. The first group is the negative group, which they don't generate any neutralizing antibody after the infection. And for the rapid waning group, the initial level of the neutralizing antibody is high. However, the antibody level drops very rapidly over the course of 6 months. And at the end of the 6 months, it dropped to a negative level. And the luckier group is the slow waning group, where the drop of the neutralizing antibody is slower. And the luckiest group is the persistent group, which have persistently high neutralizing antibody over the course of 6 months. So this study indicates that the neutralizing antibody response varies significantly between different individuals, and the prediction of immune longevity can only be accurately determined at an individual level. Many studies also investigated the neutralizing antibody kinetics among the vaccinated. First, I'm going to share the results for the mRNA vaccines, and later I will also be sharing the results for inactivated virus vaccines. As shown in this study that is now in preprint, the neutralizing antibody kinetics in the Pfizer vaccinated individuals are investigated. So as shown here, after the neutralizing antibody peak, after 1 month of the first shot, it starts to decline over the next 2 months. So and as we can see that the individual the value for different individuals is very variable. And in another study that uses the SVNT to evaluate the neutralizing antibody kinetics in mRNA vaccinated individuals for both Moderna vaccine and Pfizer. So this study was carried out for a period of 6 months. And as shown in the result here, we also observed a significant decline in average neutralizing antibody over the time. And when we look at the error bar here, it shows that there's also significant amount of variability against different individuals. For inactivated vaccine, a study from Hong Kong showed that after the first dose of the vaccine, the neutralizing antibody response is not ideal. While most of the samples still fall below the negative range. And after the second dose, there is a significant increase in the neutralizing antibody level, which is at a similar level to the first dose of Pfizer. The study from Singapore also reported a very similar finding. So this is the result for 2 doses of Pfizer, and this one is the result for 2 doses of Sinovac. So to our surprise, the results obtained from the Singapore study and the Hong Kong study is highly concordant with each other, although it's performed using different cohorts and in 2 different regions. So for the 1 dose of Sinovac, the cPass result from Singapore is 16, and the result is 15.6% for Hong Kong. And for the second dose of Sinovac, the Singapore result is 66%, while the result obtained from the Hong Kong study is 66.7%. So the value is also very close for the Pfizer vaccines. So this means that the cPass can be used as a standardized assay to evaluate protective immunity arising from vaccination. So I would also like to highlight that the variability of the neutralizing antibody level between different individuals is again shown in these 2 studies. This is especially the case for those who are vaccinated with the Sinovac vaccine. So we can see that the percentage inhibition actually can range from 90% to 25% after the second dose. So this actually highlights the need to adopt a neutralizing antibody guided booster shot regimen. So you can take a simple cPass test to know your neutralizing antibody level when you are thinking whether you should take the booster shot or not. So as shown in the graph here, the higher the cPass reading, the better. So when the value is above 68%, it's considered high titer neutralizing antibody, which is acceptable for manufacture of the convalescent plasma. And for values that's below 30%, there's no detectable neutralizing antibody. So at a population level, it is worth considering that we adopt an NAb guided booster shot regimen, that we screen out those who have low level of the neutralizing antibody and administer the booster shot for those individuals who are actually at risk of breakthrough infection. So this is also the case for those who wish to travel, and for those who need to interact with people frequently. So as the science is evolving in establishing the correlate of protection, several works have established the direct association between neutralizing antibody level and vaccine efficacy. In this recent publication in Nature Medicine, they used the international standard as the calibrator and shows that to achieve a vaccine efficacy of 80%, 26 IU per ml of the neutralizing antibody is required, while 140 IU per ml of the neutralizing antibody is required to achieve a vaccine efficacy of 90%. So we have also performed, did our own analysis based on those publications. So we have established the correlation between the range of neutralizing antibody titer in IU per ml to the vaccine efficacy for AstraZeneca or Moderna, and also their corresponding cPass percentage inhibition range. Lastly, I would like to share the cPass assay performance from the study conducted by the WHO for establishment of the international standard of the neutralizing antibody. As shown here, the blue and red represent different labs. So when performing the test for the same sample, we observe a very inconsistent result for the PRNT, which is the live virus test. So we see that for the different samples, almost all the samples have a inconsistent result in between different labs. For the cPass SVNT test, as performed by the WHO, it shows a very consistent reading that for different labs for all these samples. Again, this highlighted that cPass can be an ideal toolkit for highly standardized neutralizing antibody testing. So, in conclusion, we know that only a fraction of antibody produced post-infection or vaccination are neutralizing antibodies, and the presence of spike protein or RBD binding antibody or IgG or IgM antibody does not mean that functional neutralizing antibody are present. So as of now, cPass is the first and only FDA-approved test for detection of functional neutralizing antibody. I'd also like to share that the neutralizing antibody is a good biomarker to predict protective immunity, as shown in the evidence that I mentioned earlier. And neutralizing antibody level wanes over time, and the dynamics is highly variable between different individuals. And we also see that the neutralization activity against variant of concerns have a reduction for different at a different level according to the variant. And SVNT can differentiate such different neutralization activity. So it can be the ideal toolkit for neutralization antibody testing against variant of concerns and also pre-emergent subeco viruses. And lastly, we also observed highly concordant cPass SVNT results across different labs and across different cohorts. And I would like to end this presentation with the potential application of the cPass SVNT. Firstly, it can be applied to study the vaccine efficacy by evaluating the neutralizing antibody induction, and it can be used to identify who and when may need a booster shot. And it is also a very powerful tool for the next generation vaccine development. So that is the end of the presentation. Thank you for your attention, and I'm happy to take any question you might have. So any comments or exchange of ideas are welcome too.
Thank you, Dr. Su, for the wonderful presentation. We got quite a number of questions from the floor, so let's just answer the first question. So what are the most common causes if OD value is less than 1 in negative controls, thus not meeting the requirements and making the test invalid?
Thank you for the question. So usually the OD value for negative sample we observe is quite consistently above 1 from the different labs. There's cases that when the value rise above 1, it may be that the plate reader is not calibrated for quite some time. So in such case, I would actually recommend that you can do the test with a different microplate reader to check. Yeah, so that is one possible reason that we have encountered before.
Okay, thanks a lot, Dr. Su. The next question, as this test only detects antibody that blocks the binding of RBD with ACE2, what about the contribution of antibodies preventing uncoating of the virus post-entry?
Thank you for the question. So this is actually a very good point. So we know that the neutralizing antibody the majority of them target the RBD region, but there are also neutralizing antibodies that target other regions on the virus. But as we show in the correlation study, the correlation between the SVNT, which detects the RBD targeting neutralizing antibody, and the live virus test, which can capture all the neutralizing antibody no matter where it targets. So we still show a very high correlation between these two methods. So this means that we can capture majority of the neutralizing antibody. So because for one test we are only able to target one area. So in a practical level, the targeting RBD is the best method for a surrogate VNT. Yeah.
Okay, thank you, Dr. Su. The next question is from an attendee from India. So because in your slides, right, you have shared studies from China and also from Singapore. Is there any current or ongoing studies for India?
For Indian, actually in Indian there are quite we have customers also use this SVNT in Indian. So there is ongoing studies in India. Yeah, there are also a recent publication that from an Indian group that used the SVNT for study the Delta variant that is published in Science. So yes, there are ongoing studies that use the SVNT test for neutralizing antibody detection in Indian.
Okay, all right. Thanks a lot, Dr. Su. The next question. Do you use RBD of SARS-CoV-2 virus strain, or do you also have assays using a variant of concern? Any comparison?
Thank you very much for this question. So as I shared just now, let me go to the slide. So the RBD included in the cPass kit is based on the wild type sequence. So however, we have the option to to purchase the variant HRP RBD so that you can also detect the neutralizing antibodies against the variants. So using these variant RBDs, we have also performed studies to see the different level of neutralization against the wild type and variants. So these are the results that I shared just now using the SVNT with RBD variants. So yes, we do have studies on that. And one of the studies that have already published that you can take reference from is this one. So this one is also using the SVNT with the RBD variants. So it's published in the New England Journal of Medicine from the cPass inventor. So you can take a look at that.
Okay, yeah. Thank you, Dr. Su. So the next question, the attendee would like to ask about the conversion from percentage of inhibition to IU per ml. Do you have the method already, or do we still need to wait for the paper to be published?
Yeah, so thank you very much for this question. So this one is the result I just showed. So right now the manuscript is still have been accepted in principle. So we will see it published soon. So after it become published, we can take it as a reference to do the conversion. So yes, we will still need to wait for the publication. Yeah.
Okay, thank you, Dr. Su. The next question, what about the booster dose requirements after assessing it?
Thank you for this question. So let me go to this slide. So usually, according to the FDA's requirements on high titer neutralizing antibody for manufacture of the convalescent plasma, so for values that is above 68%, is considered high titer. If your cPass value is lower than that, it is actually recommended to take a booster shot, especially if you need to travel or if you need to frequently interact with people. So, For this value, we the higher level of the inhibition percentage, the better protection we have. Yeah.
Okay, thanks, Dr. Su. The next question. In slide 26, healthy vaccinated people have high level of neutralizing antibodies against variant of concern. How long does post-vaccination is this results?
Sorry, can I check which result is the audience referring to?
In slide 26, healthy vaccinated, yeah, correct. Healthy vaccinated people have high level of neutralizing antibodies. So how long is do they have to go through the post-vaccination to get these results?
So this study, so the days post vaccination is indicated here. So so for the Pfizer vaccine, the data is obtained after 51 days post vaccine. So for the Moderna, it's 80 days, and for Johnson & Johnson, is 39 days. Yeah. So usually, I can also share that usually the period where you develop the highest level of neutralizing antibody after you receive the vaccine is about 2 weeks to 1 month. So as I show in one of the study, so like after 1 month post the first shot, the neutralizing antibody level reach a peak level. So the usual duration is between 2 weeks to about 1 month. Yeah.
Okay. Thanks, Dr. Su. The next question, for detecting neutralizing antibodies against VOC, it's said that we just need to replace the HRP conjugated RBD with the variant of our choices. Is there any separated protocol for proceeding the cPass assay using VOC HRP conjugated RBD? Or we can just use the same cPass protocol which uploaded in the website, but just replace with the wild-type HRP conjugated RBD with VOC HRP conjugated RBD.
Thank you very much for this question. So when you can just simply replace the wild-type RBD with the variant RBD, and the protocol is still the same as when you use the wild-type RBD. Yeah. So yes, you can use the same protocol to perform the test. Yeah, okay, but take note, like if you wish to test against both the wild type and the variants, you will need to perform the test in different wells. So you will need to purchase extra other components to do the study. Yeah.
Okay, thanks, Dr. Su. The next question, SVNT can detect if you are previously infected by a certain variant of SARS-CoV-2?
This is a very good question. So, you know, as shown in this study, the SVNT can actually differentiate if you have, you know, when you test against different RBD variants, you can kind of differentiate between between the neutralization activity against the variants. So for example, if the patient is previously infected with beta, its neutralization level against beta is actually the highest. But you know, for if the patient is infected with wild type, is the neutralization level against the wild type and the alpha is very similar. So it depends on which variant you are looking at. But the SVNT is able to differentiate the neutralization against different variants, and you can potentially also differentiate whether this patient is infected with which variant. So yes, you have the potential to differentiate as shown in this data here.
Thanks, Dr. Su. The next question, can cPass percentage of inhibition represents the criteria for protective immunity from COVID-19? If yes, is this criteria been certified from WHO?
Yeah, thank you for this question. So currently, as observed in real life case studies, the cPass percentage inhibition required for required to prevent a vaccine breakthrough study, a vaccine breakthrough infection is 80% based on a populational level. So in this study, we observed that for people who have neutralizing antibody level that converted to cPass, which is 80%, they are largely prevented from the breakthrough infection. However, we all know that there's no 100% in medicine, and there can be outliers for any populational study. So the 80% can serve as a guideline to kind of differentiate whether you have enough protective immunity. So currently, we haven't yet get certified from authorities like FDA or WHO as of now. So, but we all know that the science are evolving, and we hope that one day we will be able to get this certified too. Yeah.
Thanks, Dr. Su. Okay. The next question, can cPass kit detect B cell?
Oh, the answer is no. The cPass kit is specifically for detecting neutralizing antibody, so it cannot be used to detect a B cell or T cell or other type of immune components. So it only allows detection for neutralizing antibody. Yeah.
Okay, thanks, Dr. Su. So the next question is actually a follow-up questions from the attendee who asked just now. Are the results of this neutralizing antibody test affected by lipemic samples? For example, in a patient with high triglycerides and serum appears to be turbid, will this affect the results? Is there any studies showing this?
Thank you for this question. So currently, for samples that with high triglyceride, we have tested like different samples, including those like milk sample, which also works with the assay. And for samples with high triglyceride, the assay can potentially work as well. So I think the best method is to do the test and see whether the quantity control are still within the criteria. So for our internal study, we didn't specifically test for serum containing abnormally high triglycerides, but our test, when we do our validation for the different serum, I believe that because we also didn't screen out those that contain high triglycerides, so like regular kind of serum works with our assay. And if the serum like really contain abnormally high triglyceride, you can still try with the assay and see if the quality control is still within the criteria. Yeah.
Okay, thanks, Dr. Su. The next question is
write to me regarding this and see how I can assist with it.
Okay. So the next question, which is from the attendee from Cambodia, she's asking is there's any correlation between positive neutralizing antibodies versus mortality?
Mortality. Oh, thank you for this question. So let me go back to one of the slides. So, from the summary from CDC, the level of immunity can range from, hospitalization, death, and also, symptomatic upper respiratory infection or mild disease like just cough and stuff, and also asymptomatic, infected with the virus but do not show any symptom, or sterilizing immunity, which are entirely protected from the infection. So from this different level of immunity, the T cells are mainly responsible to prevent the severe diseases such as mortality and hospitalization. And when the individual has very high antibody, especially neutralizing antibody, it can generally protect them from mild disease and also symptomatic infection. Yeah, so that is the answer to the question.
Okay, thanks, Dr. Su. The next question, can you tell us about the quantitative assay again? Like 30% is equal to how many IU per ml, 68%, and 80%, and so on?
Thank you for the question. Let me go to one of the slides. So we have actually did the correlation study, and we know that for cPass reading, if the percentage inhibition is 30 to about 60, less than 60%, the IU per ml is less than 100. And when we have actually is in the process of establishing the direct conversion of IU per ml to a percentage inhibition. So as shown in this result here. So once the manuscript is published, we will be able to do the direct conversion. So from the percentage inhibition, we'll be able to know what is the IU per ml. Yeah.
Thanks, Dr. Su. All right. We have reached 3 o'clock already, which is the time we allocated for the webinar. But let's just take on the last question from the floor, okay? So the last question. For considering the protection from infection and serious symptom, I think the combination of neutralizing antibody and CD8 activity is important. What do you think about cellular immunity?
Thank you for this question. So I would like to go back to this slide again. So cellular immunity, is likely more contribute to the protection against severe disease through a monastic response even after the antibody wanes. So we also observe cases where there's no neutralizing antibody after vaccination, but the person is still protected from from mortality and hospitalization. So it means that cellular responses are very important arm of the adaptive immunity, which plays a major role in preventing the severe diseases. While neutralizing antibody acts before the infection takes place, so it contributes to those level of immunity that is more on the mild disease and the sterilizing immunity. So this is my take on this.
Okay, thanks, Dr. Su. So we have come to the end of this webinar. Thank you everyone for attending this webinar. If you have any questions, please just write in to us and we will answer back via email later. So thank you everyone again for joining us today, and also of course Dr. Su for the great presentation. I hope everyone find this session today beneficial to your work. Thank you everyone and have a nice day. Bye-bye.
Thank you everyone. Bye-bye.
Bye-bye.